My name is Dr. Dan Goodnow, founder, Prodrome Sciences Incorporated. The title of this presentation is Plasmalogens, APOE genotype, and Alzheimer's. Just the facts. We recently published a paper titled Relation of Serum Plasmalogens and APOE genotype to Cognition and Dementia in Older Persons in a cross-sectional study. The purpose of this presentation is to dig a little more deeply into the biochemical mechanisms behind the relationship between plasmalogens and APOE genotype and their associations with dementia. There are two philosophies to keep in mind. One is Occam's razor, which basically says plurality must never be posited without necessity. And that means that complexity must be proven, not assumed. In that if you have a particular problem and there's more than one solution that satisfies that problem, we are obligated to accept that solution which is the least complicated. The second thing to keep in mind is what Albert Einstein said, is that everything should be made as simple as possible, but not simpler. So we should be equally aware of oversimplification. We don't want to simplify things to the point that they have no meaning. So first thing is the APOE genotype distributions. There are 6 possible APOE genotype allele combinations. However, most people will be one of 3. 11% of the population will have one E2 allele and one E3 allele. Most of the population will have 2 E3 alleles. The second most prevalent is individuals that have one E3 allele and one E4 allele, and there's a small group of people that have 2 E4 alleles. This is very important in that the E4 allele is associated with an increased risk of dementia. In the population presented in the paper, the distribution of the APOE alleles is as follows. So of the total population, 13% of the individuals had an E2-E3 allele combination, 64% had an E3-E3 combination, and 23% had either an E3-E4 or an E4-E4. So you can see that the distribution in the study closely matches what we see in the general population. Next, if we look at what percent of each of the alleles of the individuals with each of these alleles have dementia, and we see that 6.7% of the E2 subjects had dementia, 14% of the E3s, and 22% of the E4s. And this is pretty important because you can see a very strong linear relationship going from E2 to E3 to E4. And this linear relationship strongly suggests that there is some biochemical mechanism that is behind this observation. You'll also see in the tables much more complicated numbers. And these are basically the multivariate statistics that we use to determine if these simple graphs and simple tables are statistically robust. And the answer to that is yes. And when you see odds ratios are all relative to 1.0. 1.0 means that there is no association. The odds of having a disease or not having a disease is 1. And the lower the number is, you have a reduced risk of a disease, and the higher the number is, you have an increased risk of the disease. So here we see that the E2 allele subjects have a less than 1 odds ratio, which means they have a decreased risk of having dementia. And the E4 allele carriers have an odds ratio greater than 1, 2.1, and so they have an increased rate or increased risk of dementia. And what's important is that the positive effect of the E2 allele is approximately the same magnitude as the negative effect of the E4 allele. If we look at the plasmalogen levels, okay, since plasmalogen levels are a continuous variable, not like a genotype, which is either yes or no, we have people with low levels, medium levels, and high levels. So we have to group people into groups. And so if we group the high plasmalogen level subjects, the top 15%, and then the medium plasmalogens are around 71%, and those with low plasmalogens in that 14 to 15% range. And we do the same type of analysis as to what percent of those individual groups have dementia. We'll see that 6% of the high plasmalogen cohort has dementia, 13% of the medium plasmalogens, and 31% of the low plasmalogens. So again, we see a very strong linear relationship in that as we observe higher levels of plasmalogens in this population, we see decreasing rates of dementia. And like I just said on the APOE situation, a similar situation here is with the plasmalogen, in that this strong linear relationship suggests some biochemical mechanism behind it. And the biochemical mechanism that I'm going to discuss here today is called cholesterol efflux. And to understand cholesterol efflux, the first thing is to understand a little bit about how cholesterol is regulated. So cholesterol, we get cholesterol from our diet or from liver manufacturing, and it's transported typically in the bloodstream via your LDL protein particles. And from the LDL particles, it gets transported from the extracellular environment into the cells. Likewise, we also manufacture cholesterol endogenously in our body, and this endogenous cholesterol synthesis also gets transported into the lysosomes. From the lysosomes, the free cholesterol gets transported into the membranes. So most of the circulating cholesterol in your blood supply is in the esterified form. That's what binds it to the proteins. But the large majority of the overall cholesterol in your body is in the free form, and it exists in the plasma membranes all throughout your body. Now, when in the plasma membrane, the level of cholesterol is regulated. And how it's regulated through the esterification process. So this cholesterol can get esterified to the esterified form, and then from the esterified form, it can get picked up by HDL. And this is why the HDL to LDL ratio is an indication of reverse cholesterol transport capacity. So your higher your HDL to LDL ratios, the more efficiently you can transport cholesterol away from the cells of your body. But just like the extracellular environment transports the cholesterol, you also have cholesterol transport in the intracellular component of your cells as well. And it does the same thing. It takes this esterified cholesterol and it transports it to the endoplasmic reticulum, and it provides negative feedback loop. So when you have high levels of cholesterol in your membranes, it's leaving through HDL cholesterol transport, but it's also being sent internally and saying, oh, we got lots of cholesterol, stop making more cholesterol. And also the excess cholesterol from the lysosome also provides negative feedback to the ER. So this is how overall cholesterol levels get maintained into a certain optimal window. And so if parts of this system become disturbed, the overall cholesterol regulation and the equilibrium of cholesterol in your cells gets disrupted. Now, the APOE genotypes have a very strong association with cholesterol transport and cholesterol efflux. So this is a study done by Michikawa back in 2000, very clearly indicating that APOE proteins, generated from either the E2, E3, or the E4 alleles, have a dramatically different ability to transport cholesterol, in that the E2 has a high cholesterol efflux capacity, E3 is a medium level, and E4 is a very low level. So a very linear relationship showing how the different alleles are associated with different cholesterol transport regulations. What we also find is that in plasmalogens that we have a very similar observation. If we look at cells from children that have plasmalogen deficiencies, and these are RCDP children, RCDP stands for rhizomelic chondrodysplasia punctata. It's a rare disease in children that have a genetic mutation for certain enzymes for manufacturing of plasmalogens. And these children have very low levels of plasmalogens in their blood. So cells derived from RCDP patients look very much like an E4 carrier in a sense, in that they have reduced levels of cholesterol, HDL-mediated cholesterol efflux relative to normal cells. It was further observed by Munn in 2003 that this was due to cholesterol esterification. And she did a very extensive set of studies showing that NREL cells, which are plasmalogen deficient cells, and CHO cells, which are the normal cells, and if she was to transfect into the NREL cells enzyme vectors for plasmalogen biosynthesis, that she was able to move the cholesterol esterification from the NRELs closer to the natural cells. So indicating that cholesterol esterification was underlying the plasmalogen observation. And that this was also shown in other studies in that the NREL cells were associated with dramatically reduced levels of esterified cholesterol. And lower levels of esterified cholesterol was associated with higher levels of free cholesterol or membrane cholesterol. So this is the mechanism behind the plasmalogen association with cholesterol. Now we did a whole series of structure-activity relationships, understanding in more detail what levels of plasmalogen, what type of plasmalogens were associated with this cholesterol esterification observation. And what we found was that increasing levels of DHA plasmalogens, or the polyunsaturated phospholipids, resulted in a dose-dependent increase in the sterol O-acetyltransferase activity levels. And that other compounds like pravastatin that are HMG-CoA inhibitors had no effect on cholesterol esterification enzymes. And what we saw was that with increasing levels of DHA plasmalogens, we found an increased levels of esterified cholesterol. And this increased level of esterified cholesterol was associated with a dose-dependent decrease in free cholesterol. So here we have a very simple causal relationship of how plasmalogen levels are associated with and actually can drive cholesterol levels in cells. So overall cholesterol transport, the two systems, so plasmalogens are affecting cholesterol transport by modifying the esterification process. So by higher levels of plasmalogens, we can push more cholesterol out of the cell through esterification. The APOE genotypes, on the other hand, they affect how this cholesterol, once it leaves the cell, can be transported. And so the E4 allele proteins have a very poor capacity, so it can't really pull much cholesterol out of the cell. Whereas E3 carriers can pull a medium amount, and E2s are very efficient at moving cholesterol out of cell membranes. Now, there's a very complicated table in the paper called Table 4 that looks at the multivariate associations of clinical and biological variables with serum lipids. And let me take a couple minutes to explain this table in simple language. So the APOE3 is the reference. So if we want to compare E2 to E3 and E4 to E3 in these different levels, LDL and HDL, the key thing to observe is that the APOE effect is really being mediated by the LDL cholesterol levels, in that the E2 allele is associated with lower levels of LDL cholesterol quite significantly, whereas the E4 allele is associated with higher levels of LDL cholesterol. The APOE genotypes have no statistical effect on HDL cholesterol at all. So when we look at the ratio of HDL to LDL, we see that the E2 carriers are associated with a high HDL to LDL ratio, which is an indicator of reverse cholesterol transport, whereas the APOE4s are associated with a lower HDL to LDL ratio. It's important to note that biological systems try to maintain levels of equilibrium. So if the E4 carriers are having a difficult time moving cholesterol in reverse cholesterol transport, that means that there's more cholesterol there in the free form, which will cause LDL particles to basically expand to meet that equilibrium. Now, plasmalogens are also associated with a higher HDL to LDL ratio, very much like the E2 carriers. But the difference in the plasmalogen observation is that it's mediated by HDL cholesterol, not the LDL cholesterol. So plasmalogens are associated with high levels of HDL, and it's also associated with higher levels of LDL. But the increase in HDL is much higher than the increase in LDL, resulting in the HDL to LDL ratio being significantly increased in the high plasmalogen subjects. The other two things to note is that high levels of plasmalogens are associated with low levels of triglycerides. And this is because plasmalogens are manufactured in peroxisomes. And peroxisomal function is associated with low triglycerides because peroxisomes, through their beta oxidation mechanisms, consume lipids. And the final thing is that you'll see that high levels of plasmalogens are associated with actually higher levels of overall cholesterol. And this is also consistent with increased peroxisomal function in that the acetyl-CoA generated from peroxisomal beta oxidation is what the body uses to manufacture cholesterol. So all of these numbers are very consistent with known biochemical observations in the human body. So next step is, okay, so membrane cholesterol efflux and membrane cholesterol levels are associated with plasmalogens and APOE genotype. So what is the deal with membrane cholesterol? Well, membrane cholesterol itself is associated with the severity of dementia. So this is a paper from Cutler in 2004, and he looked at postmortem human brain tissues, and he showed that the cholesterol, not overall cholesterol, but just membrane cholesterol levels, okay, individuals that had severe dementia versus moderate dementia versus mild dementia, okay, had higher levels of free cholesterol in their membranes. So high levels or increased levels of free cholesterol in the membrane of the brain is associated with higher or a higher degree of cognitive impairment. Very similar to what we're discussing. Now, the next part about membrane cholesterol levels and membrane cholesterol regulation is the association of membrane cholesterol and amyloid formation. So amyloid, which is of course one of the hallmarks of Alzheimer's disease, and it's increased in E4 carriers, is a product from a precursor protein called APP. This is amyloid precursor protein. And APP is processed by one of two enzymes, either beta secretase, which cleaves APP at a certain point, and it results in A beta 42 fragments, and these are the fragments that go on to form the amyloid plaques that are the pathological hallmarks of Alzheimer's disease. Alpha secretase, on the other hand, cleaves APP at a different point, resulting in a product called soluble APP alpha. When APP is processed by alpha secretase, we do not get pathological plaque formation. So alpha secretase is healthy APP processing, and beta secretase is the pathological APP processing. Why this is important is that these two enzymes prefer, and they exist in different parts of your biological membranes. So beta secretase prefers to live in what's a region called cholesterol or lipid rafts. And lipid rafts are areas that have high levels of cholesterol and high levels of phospholipids with saturated fatty acids on them. And make a note to remember this flotillin. Flotillin is a protein that's associated with lipid rafts. It's a biomarker of lipid rafts. Now, alpha secretase, on the other hand, it likes to be in the phospholipid-rich membranes, and specifically in phospholipid-rich regions with unsaturated fatty acids. It turns out that if we manipulate or modify membrane compositions, we can modify APP processing, and so that we can actually increase or decrease A beta 42. And so we published this back in 2011. And so if you take membranes and you add cholesterol to them, adding cholesterol results in an increase in beta secretase. And if we add DHA plasmalogens, which is a polyunsaturated phospholipid region, we dose-dependently increase alpha secretase levels in the membranes. And when you do that, you can directly manipulate A beta 42 formation. So A beta 42 being the protein that forms the amyloid plaques. So if we add cholesterol, we increase beta secretase, we increase A beta 42. We add DHA plasmalogens, and we decrease A beta 42 formation in a very dose-dependent manner. So here you have a very simple causal relationship between membrane composition and amyloid formation. The question is, does that observation in cell culture in the laboratory, does that actually happen in humans? And the way we can find that out is that we look at post-mortem human tissue. And so we studied 100 individuals from the Rush cohort, okay, that had no cognitive, like 38 had no cognitive impairment, 24 with mild cognitive impairment, and 38 with Alzheimer's dementia. And of these individuals, 10 of them had APOE 2 allele and 3 allele, 51 were E3s and E3s, and 31 had either E3, E4 or an E4, E4 allele. And we looked at a whole bunch of other things. But first and foremost, let's talk about cholesterol and, well, not cholesterol, let's talk about beta amyloid. So as you'd expect, okay, non-E4 carriers had low levels of amyloid in the brain, and E4 carriers had high levels of amyloid in the brain. Likewise, people that had high levels of DHA plasmalogens in the brain also had low amyloid in the brain, and people with low to moderate levels of DHA plasmalogens had high levels of amyloid in the brain. More importantly, E4 carriers, okay, with high levels of DHA plasmalogens actually had normal levels of amyloid in the brain. So here we're seeing in human postmortem brain tissue exactly what we're seeing in laboratory cell culture, in that both the E4 and the DHA plasmalogen observations are observed in humans. Now, when we put all this together, we want to look at how does it all make sense when we look at the interaction map. And what this means is that the APOE4 genotype, okay, is associated with membrane dysfunction. And that membrane dysfunction further results in amyloid, but not all E4 carriers. So what this graph shows, if our outcome is reduced cognition, this says that yes, E4 is associated with cognition, but it has its association through amyloid. So it's only the E4 carriers that have high levels of amyloid. So once you know the amyloid level in individuals, the genotype is irrelevant. And the next step along the pathway is tangles. So amyloid is associated with reduced cognition, but if you know the tangle density in individuals, you don't need to know the amyloid. So it's only individuals that have amyloid, so people with high levels of amyloid, okay, it's only those that have high tangle density that have reduced cognition. So it's not the amyloid, it's the tangles. And when we do all this work, we found there's 4 things that were critical: the DHA plasmalogens in the brain, tangle density, the choline high affinity transporter, which is essentially a biomarker of cholinergic neurons, and flotilin, which is a biomarker of lipid rafts. And we look at the odds ratio, we see that the strongest association is the DHA plasmalogen, so 0.2, which is the same as a 5-fold effect. So 1 standard deviation change in DHA plasmalogens in the brain was associated with a 5-fold change in dementia. So 1 standard deviation increase resulted in a 5-fold decrease in dementia. Flotilin was the opposite. So increasing flotilin levels means increasing lipid raft area, means increased risk of dementia. Choline high affinity transporter, which is a marker of cholinergic neurons, basically says increasing cholinergic neuron density results in a decreased risk of dementia. So more cholinergic neurons, the better. And tangles is the opposite. The more tangles you have, the higher your risk is to have dementia. So it all makes simple sense. So in the end, if you take a look at table 2 of the paper, we can ask the question, what's the the dementia rate in the different genotypes according to their plasmalogen levels in their blood. And we get a very interesting thing. So in the E4 carriers, we see that people with low plasmalogens had a 37% rate of dementia, moderate plasmalogens 20, and high plasmalogens 14%. So you can see very clearly we get almost an exact 7% decrease in dementia rate with each standard deviation increase in plasmalogens. And we see the same effect in the E3 carriers, in that we go from 30% to 12% to 6%. And in the E2 carriers, we go from 22% to 6% to 0. And what's interesting here is that the low effect is pretty dramatic. Now, of course, this is an observational epidemiological study. So we are restricted by the levels of plasmalogens that are naturally occurring in individuals. So we have no way of knowing what the effect of high levels of plasmalogens are that are higher than normal levels. And that's what clinical trials are required to do. So the real question for the APOE genotypes is that this relationship from 37 to 20 to 14, does it continue? Okay, if we go to 2 standard deviations above the median level or to 3 standard deviations higher, can this 14% get pushed down to 6% and down to 0%? And the only way to understand that or to prove or even to investigate that is to run clinical trials with plasmalogen supplementation, which is what we're designing. So that, in summary, is an overall presentation of plasmalogens and the APOE genotype and the relationship to cognition and dementia. And of the available evidence to date, on the APOE side, the cholesterol transport process is clearly the most obvious biochemical mechanism. Now, plasmalogens have a secondary role because they're also involved in vesicular transport and membrane fusion activities, and that was presented in a different one of my presentations. But hopefully this provides some additional context to the APOE4 community in regards to your risk of dementia associated with your genotype. Thank you very much. Bye-bye.